Well, first of all, thank you very much for hosting me here. And the subject of our conference, the title of our conference is How to. So today I'm going to talk about how to use spiders, female spiders, to understand the Parkinson and Alzheimer disease. So I'm quite fascinated about the history of Alzheimer's disease discovery, and I would like to tell you this history. Imagine yourself in Frankfurt in November 1890. A young, just graduated doctor, his name is Alois Alzheimer. He decided to earn a bit of money on the side, and he accompanied a woman. She was at that time a wife of wealthy men, and she was traveling from one town to another, and she was not feeling well. So he accompanied this woman. He served basically as a general practitioner. He recorded her symptoms, he did his job, got his payment, and continued with his career. 15 years later, the same woman arrives to psychiatric hospital in Frankfurt. And she arrives with weird symptoms. She has difficulties to identify herself in time and space. She has difficulties to recognize her relatives. And she has difficulties in all aspects of social behavior. Alzheimer immediately got curious about her case, and he decided to observe her social behavior and the physical symptoms. And after her death, which happened just a few months later, he performed a pathoanatomical analysis of her internal organs. So what he found out, he found a relatively large clumps in her brains. And those clumps were made of protein fibers. And these protein fibers, the presence of these protein fibers basically destroyed the neuronal cells in her brains and has led to her death. Since then, since the discovery of Alzheimer's disease, by the way, the name of the disease was after this German psychiatrist, enormous efforts from the researchers all over the world have been directed toward the understanding the reason what drives the normal protein to misbehave and suddenly to decide to form these toxic fibers, namely amyloids. Amyloids is a common name for the protein fibers that are usually lead to neurodegenerative disorders, whether it is Parkinson or Alzheimer. So I'm a chemist and from Weizmann Institute of Science, and I'm in quite fortunate position to run my own research lab. And running a research lab is like managing a non-profit startup company. And what we are doing in our lab, we are trying to answer a series of big scientific questions. The first question is why actually protein decide to misbehave, at what conditions, and what is the mechanism of protein fibrillation, how we can stop the protein fibrillation, and what is the similarities and differences between the toxic amyloids, toxic fibers, the fibers that lead to neurodegenerative disorders, and beneficial fibers, the fibers that are produced by spiders and silkworms. And what can we learn from spiders and silkworms and the cure of Alzheimer? So what is the question, or what is unclear? Here you can see two female species. The first one is Augusta Detter, the same patient that was identified with the Alzheimer's disease. Oh, it's here. Okay. She was identified with the Alzheimer's disease in 1902. And on your left, on the right, sorry, is Amorobius similis, the female spiders. Both of them are producing an enormous amount of fibril proteins, protein fibers. However, in the case of Augusta, these fibers are quite toxic, and actually the presence of these fibers in her brains was the major reason for her death. In the case of a female spider, the fibers are quite beneficial because without these fibers, he cannot survive. And what is interesting is if we take a sample from both type of spiders, whether it's amyloids or whether it's silk, and examine them under the most powerful microscope, like atomic force microscopy, the pictures that you can see here, we probably won't find any difference. Moreover, if we go forward and perform an X-ray analysis, the crystallographic analysis that will tell us information about the distances between the crystals, one crystal in a protein fiber and another crystal, and these distances between these structures, we also probably won't find any difference. Both of them are driven by the hydrogen bond formation. However, we know little about the amyloids, and we know a lot about silk. And apparently, being so similar to amyloids, silk is quite popular material. Where silk is coming from? Silk is coming from these guys. Those are silkworm cocoons. They are produced by Bambux's moray silkworm. Bambux's moray silkworm is an industrial producer of the silk, and apparently this silkworm is not only the producer of silk protein in industrial quantities, it's also a creature that is quite easy and susceptible for genetic modification. So what we are doing, when we are taking the silkworm, inserting the genetic information, and forcing the silkworm to produce a spider silk for us. Silk is quite ancient material, and it has been around for already 5,000 years and contributed enormously to the development of worldwide economy. And the standard silk-based technology is, of course, textiles. Silk also has been used in non-standard based technology, whether it's surgical threads that are biodegradable and tend to dissolve while it's exposed to our body fluids. Silk protein has an exceptional mechanical properties, the silk fibers, and also use an additive to improve the mechanical properties of the existing material. For example, we are using silk to improve the mechanical properties of rust-free panels in the boats, or we are using also silk to improve the mechanical properties of a protective case for our iPhone 7 Plus phone. Silk also used in nanotechnological application, whether it has been converted into the threads, microspheres, films, microcapsules, foams. Silk, however, in all these applications, we used silk rather as an inert material, meaning there is no biological function. What we know about the silk? Silk is produced by different insects, whether it's spiders, silkworms, scorpions, honeybees, ants, flies, bugs, and has a unique mechanical properties, is biodegradable. Our body knows how to deal with this, and silk is a material that among the first being approved by FDA. It's transparent material, therefore we can use the silk for optical devices. We know the structure of the silk. However, what we don't know, silk has been extracted directly from the organ where the insects store the silk, a silkworm gland, for example, is highly sensitive and aggregation-prone material. So silk tends to aggregate and to form fibers within a second. And silk also shares structural similarity with toxic amyloids. It's almost identical. So the next question is whether we can use silk to get rid of the toxic amyloids. And the answer is, or in other words, can we use nature to heal nature? And the answer is, we hope so. In order to be able to do this, we need first to understand or to overcome several challenges. First, we need to understand the differences and the similarities between the silk and amyloids. Apparently, in both cases, whether it's toxic fibers or whether it's silk, in both cases, at the atomic level, the formation of the fibers is driven by the formation of the hydrogen bonds, or intermolecular or intramolecular. The same hydrogen bonds, they are responsible for making our drinkable and transparent liquid as a liquid, like water. And as soon we break these hydrogen bonds by, for example, applying the heat, by heating the water, these hydrogen bonds are relaxed and broken, and the water is converted into the gas. So these hydrogen bonds are forcing the protein to refold, to lose its three-dimensional structure, and to form crystals. Those crystals aligned on the top of each other, forming the fibers. And in the case of silk, the bunch of fibers are spun into the cocoon that you have already seen. So what is the difference between the silk and amyloids? Apparently, the major difference is in the distances between these crystalline regions. In the case of amyloids, of the toxic amyloids, the distance is too small and the crystals are packed too tight to each other that our body has no tool to come and to cleave it, to cut this bond and to digest these fibers. In the case of silk, the distances between these crystalline regions is way larger. So our body can send a soldier molecules like an enzyme and cleave the bonds and digest the silk fibers. Therefore, silk fibers are biocompatible. So now we know more or less the difference between silk and amyloids, and the difference is in self-assembly pathway or in the formation of the protein fiber, mechanism of formation of protein fibers. So the next rational step is to try to control these self-assembly processes. Animal or insects like arthropods or silkworms are doing this by simply regulating the amount of water and the amount of protein. More hydrated the fiber, less crystalline it is. As soon we dehydrate the silk fiber, we induce the additional crystallization, and the fiber is finally converted into the solid and quite stable and mechanically tough, strong fiber. How we are controlling self-assembly process in our lab. So we are basically using 3 types of silk. The first type is native silk, the same silk which is directly extracted from the organ where the animal store its silk. It's a silk gland. We simply cut the gland and extract the solution which is stored inside the gland. And this kind of solution is quite precious. It has pharmaceutical or bioactive properties. And the parameters that we can control upon the extraction of the silk from the silk gland is basically none of them. We are highly dependent in this case from the insect. Basically, the insect dictates us what will be the final mechanical or pharmaceutical properties of the silk, of the soluble silk. The second type of silk is the same silk that we extract from the silk cocoon by simply, first of all, unwrapping the cocoon by boiling this in water and detergent, and applying additional chemical resolubilization by boiling the cocoon in strong chemicals like lithium bromide. And in this case, we can play with the ratio of protein subunits. Apparently, silk consists of 2 protein subunits, the large subunit and the small one. And playing with the ratio of heavy and light chain in the silk, we can also change the mechanical properties of the fibers. And the third type of silk is a silk produced by the bacteria. We simply take the body of the E. coli bacteria, insert the genetic code or genetic information, and the bacteria produce silk with the properties we want for us. And in this case, we can control basically everything, whether it's sequence, genetic code of the silk, three-dimensional structure conformation, fibrillation process, mechanical and pharmaceutical properties of silk. This is what happened on the single molecule level. How we control the silk on a sort of three-dimensional or micron scale structure. We are using a technology called microfluidics. Well, I'm not going to explain deeply the principle of microfluidics. I will show you just a microfluidic chip. What is microfluidic chip? It's basically a chip made of silicon. And inside the chip, we have channels. And inside these channels, we basically we flow our solution, chemical solution, and we manipulating the flow. So it's like in our veins. By manipulating the flow, we are able to break the protein solution into the droplets or into the nanoscale particles and produce various of shapes. We are able to control inside these channels shear stress, chemical environment, fluid flow, and these parameters has a direct implication on the shape, size, and protein conformation of our protein locked inside this structure. And apparently this structure in the future could be used as a delivery agent to deliver our components to the targeted organs. Yes, now we know how to control self-assembly process. It depends on what type of protein we are working with. We can either go the way which insects are going by regulating the amount of water in the structure, or by changing the protein structure via inserting the genetic information into bacteria cell, or by simply structuring proteins into biomaterials. The next challenge is quite tricky, how we can stop self-assembly, and apparently with the main emphasis on self-assembly of toxic amyloids. And it's quite hard, because silk itself indeed has ability to bind to amyloids. When we mix silk and amyloids, silk binds to amyloids and stop the further growth of the fiber. However, if the amylogenic fiber is already formed, silk is not able to dissolve it. So we can stop the aggregation, but we cannot sort of get rid of the fibers that are formed. And what or who can? Apparently, there is an animals who has a certain molecules that has a capability to dissolve these fibers. And those animals are camels and llamas. And the molecule which is responsible for dissolving the fiber called antibody. And to be more precise, this part of antibody, nanobody. So we can make use of this molecule to dissolve already existing fiber. Now, the next question that you probably have in mind, why we need silk? We can simply inject this antibody or nanobody. It will dissolve the pathological fiber, and that's it, we solve the problem. Apparently, these molecules are super sensitive, and it tends to dysfunctionalize upon the exposure to the same conditions under which amylogenic and toxic fibers are formed. And we need some helper molecule to stabilize it. So what we are doing in the lab, we are trying to test the effect of silk on the aggregation pathway of amyloids. In the classical sort of mechanism, amyloids are forming via forming first of all the nucleus of the protein, and then this nucleus serves as a triggering point for fiber growth or fiber elongation. Apparently, if we mix amylogenic protein with the silk, we observe the formation of the first nucleus, but in this case, the fibers, the mature fibers, were not formed. And when we created the complex between the silk and a nanobody, we haven't observed any nucleation processes and no fibril growth. So to stop self-assembly processes in amyloids, we either can use silk in the case that amyloid fibers are not formed yet, or a combination, a complex between the silk and existing therapies. So I would like also to use this opportunity to thank our group members, Tom Mason, which is responsible for microfluidic part, Dror Elias, Jenny Lee, Nidhi, and Elad. Well, and thank you very much for the attention.